Welcome to Longevity by Design, a podcast designed to give individuals access to the leading scientific information in the field of longevity. The ability to add years to your life and life to your years needs no opinion. Join us as we ask science to take the wheel. In each episode, Dr. Gil Blander joins a co-host and an industry expert in the field of longevity, shining a light and getting the answers to the key question: how can we live a longer, healthier life?
Hello, I'm Ashley Reaver, and I'm joined by Dr. Gil Blander. Welcome to Longevity by Design, How to Live a Longer, Healthier Life. We're produced by Insight Tracker, your science-based guide to optimizing your body from the inside out. Our guest today is Dr. Tony Wyss-Coray. Dr. Coray is the D.H. Chen Distinguished Professor of Neurology and Neurological Sciences and the Director of the Phil and Penny Knight Initiative for Brain Resilience at Stanford University. His lab studies brain aging and neurodegeneration with a focus on age-related cognitive decline and Alzheimer's disease. The Wyss-Coray research team discovered that circulatory blood factors can modulate brain structure and function. They have also found that factors from young organisms can rejuvenate old brains. Dr. Wyss-Coray was voted Time Magazine's the Healthcare 50 Most Influential People Transforming Healthcare in 2018. He co-founded Alkahest Inc. and several other companies targeting Alzheimer's and neurodegeneration, and has been the recipient of an NIH Director's Pioneer Award, a Zenith Award from the Alzheimer's Association, and a Nomis Foundation Award. Thank you so much for being with us today.
Thank you. Thanks for having me.
So again, Tony, it's a pleasure for us to have you. And we usually start by asking our guests a question about what made you to become a scientist, and how have you drifted from your initial goal to study longevity and aging?
Yeah, that's a great question. I was always fascinated by nature, and grew up in a relatively rural area in Switzerland, and we had a big garden, and I wanted to become a farmer actually when I was little, and then as I got older, started to discover science and collected plants, and was really fascinated by nature. And I think that's how I then ended up going to university in Bern, actually. And we had an interesting track that provided exposure for a semester to zoology, botany, microbiology, immunology, and biochemistry. And so you got sort of a broad overview in the first 2 years of undergrad. And I ended up going towards immunology.
Amazing. And from there, found your way to longevity and neurology?
Yeah. So how did I get there? I did a PhD then in immunology. A basic T cell antigen presentation is what I studied. Also at Bern. And then I knew I wanted to study the immune system in vivo. And it's interesting that immunologists have often used the brain to study the immune system. Multiple sclerosis, for example, or viral infections of the brain provided key insight into our immune system. And so I looked for labs in the US. I knew I had to go to the US and work in a top lab to have a career in science, but it wasn't my plan to stay here. So I ended up in San Diego at Scripps with Leonard Mukey. He's now the director of the Gladstone Institutes of Neurological Disease in San Francisco. So I joined his lab, and then 2 years into my postdoc, he was recruited to San Francisco to start the Gladstone Institute for Neurological Disease there. And so for us, it was either, staying there at Scripps and find a new lab, or going back home, or move with him. And so we decided to move up to the Bay Area, to San Francisco. And I was at the Gladstone then for 7 years at UCSF before joining Stanford faculty in 2002. But the way I got into Alzheimer's disease and aging is really initially studying the immune system in the brain. We expressed regulators of the immune system for the first time in mice, and could then observe all kinds of effects on um, neurodegeneration and inflammation in the brain. And then Leonard, my boss at the time, started to help develop the first mouse models for Alzheimer's disease. And so we crossed these mice that had immune modulators with mice that modeled Alzheimer's disease and were able to manipulate basically the immune system in these brains of so-called Alzheimer's mice. And it turned out that that had huge effects on the development of the disease, either promoting it or slowing it down. And over the years, then I got more and more interested in, why do people actually get this disease? And why is it always age related, right? You don't get Alzheimer's disease when you're young. And so I got more and more interested in aging. And then also, in studying humans, because these models turned out to have relatively limited utility or, for understanding how the disease actually develops, because we induced it in an artificial way in these mice. And it's still useful to study some aspects of the disease. But ultimately, if you want to understand how the disease develops in humans, you have to study humans.
Sure.
Yeah.
Okay.
And Tony, a question related to what you said before. You came and your goal was to go back to Switzerland. And I have actually the similar story. I came and I wanted to go back to Israel. So what made you to decide to stay in the US and not move back to your home country?
Yeah, it's, I mean, I had the luxury of getting a fellowship initially for 1 year, and then I renewed it and got 2 additional years of funding. So, it was relatively easy. And I had actually, from Switzerland, I got a pretty decent salary for a postdoc. I think I had much more than my fellow postdocs who had money from the US.
Yeah.
And my wife, she's a nurse, and she was pretty open to stay there. San Diego is beautiful. We went to the beach, we traveled around on weekends, and we really liked, the environment. I mean, it's so beautiful there. But then also coming up, we decided, let's try it and see how is San Francisco. We actually got some support from our parents to buy a house in Berkeley. Now, for a ridiculously low amount of money, if you look back. But, at the time, it was anything we could afford. It's a very small house, 1,000 square feet, in the Berkeley Hills, but was beautiful. And, we started to make friends and life was good. So I think we really liked to live there. It's a very, beautiful environment, very liberal, people from all over the world. But then also scientifically, the opportunities were just amazing. And because I switched field from immunology to neuroscience, I didn't know anybody in Switzerland. It's a very small country. So I think it's very similar to Israel, right? If you switch a field and you don't know anybody there, it's pretty hard to find a position. And so I applied a few times, but didn't get a good position. And, I started to be very picky where I wanted to go. So it was, maybe Basel, had a couple positions, the Friedrich Miescher, or then ETH is really, or University of Zurich, where I would have had gone. And, then the opportunity in Stanford came around, and I kept on, getting offers, and I actually got a really very good offer from Switzerland about 7-8 years ago, but then it was almost too late. And
Yeah. I hear it a lot from an immigrant, and I felt it for myself. You have a window of opportunity, but then when the kids get older, the wife get
Yeah.
more, you get
Yeah.
more settled down. It's very hard to move back. So it's
That's right.
literally a window, a very small window of
That's right.
opportunity
Yeah.
that you can
Yeah.
go back.
That's exactly right. Yeah.
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 Okay, good.
Yeah.
Thank you
And my
so much.
wife also, she works on, with Alzheimer patients, actually. She works in a memory clinic. And initially with Bruce Miller at UCSF and now at Stanford at the Alzheimer's Research Center as a coordinator and sees patients. And she really liked it. And so, as you said, both of us had good careers, and then the kids, also, they liked it here. I mean, we always went back, and, they really love Europe. It's interesting, when we go to Europe, they defend the US, and when we're here, then they defend Europe. You probably
Bay
have to
Area is a
say,
nice place to land.
Yeah.
Yeah, it's true.
Well, a few things before we kind of dive deeper into maybe some of the sciences. You are the founder of 3 biotech companies, Alkahest, Noto, and Teal. Would you mind sharing a bit of info with us about what they are? Maybe describe their origin and what they do.
Yeah, so they really, I mean, they all emerged out of our research and sort of, naturally in a way. I had a previous company that didn't really work out, but that also started by, somebody approaching me and proposing to start a company. And that was actually a biomarker company for Alzheimer's disease, but we were way too early. Didn't work out. And the nice thing in the US, which is a bit different from Switzerland, is when you fail, people say, it's good, you learned something and you should try again. The next time will be better. And that's not exactly the way I grew up. People, usually point, I told you it's not going to work or something like that. So then, I had the opportunity to get to know Caroline Nikolic, who's, a serial entrepreneur. He actually started the neuroscience program at Genentech. And he approached me, he had connections when one of our papers came out that showed these rejuvenating effects of blood on mouse brains, on old mouse brains. He basically found an investor out of Hong Kong who wanted to start a company. And so, he called me one day and said, hey, you have this, person who would like to see the company. Are you interested? And it's, sort of surprised. Yeah, why not? And then, a few months later, he called me again and said, okay, it's for real. Do you want to do it? And so that's how Alkahest got started. And then Kinoto came more out of really IP that, as we made discovery in the lab, findings that we made that seemed like good material to start a company around the blood-brain barrier and also lysosomal function. And then the most recently, Teal again came out of our proteomic analysis, and I'm sure we're going to talk a little bit more about that. Sort of changes that happen in the composition of the blood in people as they get older or develop disease. And it sounds like a very exciting opportunity. And as you get more experienced, I think, and have experience with previous company, I think, investors tend to have more comfort in giving you money and starting something. And so it becomes like a bit easier. And right now, probably I don't have any capacity to do anything else or start another company. And I have no desire. I really want to get the ones that we have to the next level. And I should mention Alkahest has actually been acquired by Grifols. So I'm still an advisor there and involved in sort of what happens, but it's much more, going towards clinical development.
Congrats on that.
Thank you.
Yeah. And Tony, we met last time at the ARDD meeting in Copenhagen. And you provide an amazing presentation there about proteomics and organ-specific aging. So I think that the majority of the discussion today will dedicate to that. You show data on a cohort of around thousands or tens of thousands of people with a 15 years follow-up, and you assess like around 5,000 proteins to calculate risk of mortality, organ aging, and so on. So I would like to start by in the basic and basically explain to our audience what is proteomics.
Yeah, proteomics is a term that people use when you measure many different proteins, usually thousands of proteins in a sample, in a so-called biospecimen. So that could be a blood sample from a person, or it could be cerebrospinal fluid. It could also be tissue. But the term omics people add to methods or molecules when they do a lot of them. You could also say lipid omics when you measure lots of different lipids, for example.
And you during your study, you use a technology called SomaLogic or the SomaLogic platform. So can you try to explain? I know that it's very complex, and I'm not sure that all our audience will understand it. But what is SomaLogic, and what is the advantage of such a platform?
Yeah, so the way you do it, if you want to measure thousands of proteins, you need, of course, very sophisticated tools. The field had developed methods to measure one protein at a time, but over the past 10, 20 years, methods were developed that really can measure these thousands of proteins. One is actually with an older technology called mass spectrometry, where you can measure any type of molecule really based on the method you develop. But these more advanced or more specialized tools like SomaLogic, you mentioned, they basically develop, if you think about a protein as sort of a three-dimensional structure, they produce a molecule that can recognize that structure. It's like a lock and a key. And they produce that for thousands of different proteins. And it actually allows you to measure these proteins in just a drop of blood. So it has really the advantage that you don't need a lot of blood from a person, and you get thousands of different measurements. And why would we want that? We want to really understand biology in an unbiased way without having a preconceived idea what we're going to find. And so the more of these proteins we can measure, our body has about 20,000 genes, and from each gene, you can make one of these proteins. And the more of these you can measure, the more you learn about the biology or the disease in a person. There's another platform from a company called Olink, and they can also measure several thousand. They can now measure 5,000 proteins, and they use a slightly different technology, but it's based on the same idea that you have these basically tools that can specifically recognize different proteins. And these proteins could be hormones, they could be, something like insulin, or you may have heard of interferon or TNF. So all kinds of different building blocks of biology.
And the research that you presented on had a very long follow-up period, 15-year follow-up period, which is super significant. What were some, or can you share some of those main discoveries from this experiment, especially those that are related to aging?
Yeah, absolutely. So maybe to just mention the cohorts that we have, we call these cohorts. So these are samples from groups of people who volunteer for research usually. They come from Alzheimer's research centers at Stanford as well as Washington University. And also a very interesting cohort from near Barcelona at Einstein College of Medicine in New York. He studies centenarians and their children, and then unrelated, the spouses often, and then unrelated people also to some extent. And from that study, actually, from NEAR is where we had the 15-year follow-up. We didn't have that much follow-up from the other studies that we had. So, as you said, that really provides you the possibility to get a blood sample from somebody at time 0, and then 15 years later, you can see, is that person still healthy, or did they develop a disease, and what type of disease did they develop? And so what we found is that we can basically measure the relative age of organs in your body, of, let's say, the heart, the liver, the kidney, the brain, simply based on the composition of the blood. So when we measure these thousands of proteins, we find that some of them come from, for example, are being produced only in the kidney. So if we then measure these proteins, they tell us something about the kidney. Similar to when you go to the doctor takes some blood and measures, let's say, insulin. They can tell you whether you have diabetes or not. Or if you measure prostate antigen, whether you have maybe an enlarged prostate or maybe you have cancer. These are all basically measurements that a physician will do in your blood that allows them to tell you whether you're healthy or not. And so we use that same idea, but we look again at many more proteins that your regular doctor would look at. And we look at proteins that are produced, they're present in the blood, but they're produced in the kidney, for example, or in the heart, in the brain. And so if they change in levels, they tell us something about the function of that organ, whether it's function normally, but it also tells us something about the age. So what we could do with this tool then is we were able to predict or measure basically the biological age of all these major organs in a person. So if you gave me your blood, I can measure these proteins and I can tell you approximately how old your body as a whole is, but more interestingly, how old is your heart? How old is your brain? And what we have discovered, not just our lab, but the field now, is that, let's say you're 50 years old. It turns out some of your organs are a little bit older, not 20 years older, but they may be a couple years older, and others may be younger. And they don't all age completely in synchrony. And what the field is proposing, and increasingly there is evidence that this is the case, and some from our lab, from the study now, is that if your kidney, for example, is older, you're at higher risk to develop kidney disease or diabetes, for example. If your heart is older, you're more likely to have a heart attack. And especially in that 15-year follow-up study, we could show, first of all, without knowing, we could say who had a heart attack because their proteins had different levels in the blood. But more interestingly, we could predict who is likely going to have a heart attack, not with absolute certainty, but with a higher certainty than most other measurements that we have currently in medicine. For example, if you're overweight, you have a certain risk to get a heart disease, right? Or if you smoke, all these are risk factors that everybody probably knows, who everybody knows. And so if if we now have higher confidence to know who actually gets the disease, the more precise we can make that prediction, the better we can tailor drugs, for example, or really convince a person to do something about their health, right? If you say somebody, oh, there's a 5% chance that, you know, you will have a heart attack, maybe they take that chance and they don't change their lifestyle. But if you tell them, if you don't do anything in 2 years, you have a 50% chance of a heart attack, maybe then they wake up and say, okay, I'm going to change my life. If they can, of course.
So couple of comments. First, we interviewed Nir Barzilai around 2 years ago. So anyone that is interested to learn more about the cohort that Tony used, you are welcome to look back and listen to Nir. It was a fascinating discussion.
Very fun. I laughed a lot in that too.
Yeah.
Oh,
telling
yeah.
stories.
I mean, he's a stand-up comedian. He's just incredible.
He is. And the second point, which I think that it's very in line with what, in a way, InstaTracker is doing, is prevention. And what, Tony, what you found, if I'll try to describe it, and you're more than welcome to correct me, is the platform that you build provides some risk assessment for a user based on a big proteomics, but that's not your destiny. That's basically the risk assessment of today. And you can change your behavior. And hopefully based on that, you might not have this risk or bad car that you have might not become a reality. So don't be discouraged by that, but try to fight it and prevent it.
Exactly. Yes. Yeah. And, you know, this is what so-called precision medicine would like to achieve, right? That on an individual basis, you can tell somebody what their relative risk is to get, you know, dementia or heart disease or, you know, diabetes or cancer. And most of these have an aging component. You know, most of these diseases we get when we're older. And so it seems that if a given organ in your body ages a bit faster, you are more likely to get a disease of that organ. So if your brain ages faster, for example, you're more likely to show cognitive decline, especially people who are already complaining about memory problems. And we call this mild cognitive impairment, for example. So they have already some signs of memory problems, but they don't have dementia yet. So they can still, you know, you can still have a conversation with them, but they may be quite forgetful. So in such people, if they have a significantly older brain with our method, then they're much more likely to decline over the next 5 years than somebody who has a young brain.
Yeah. And that's taking us back again to Nir Barzilai somehow. Nir is now trying to push aging as a disease. And I know that you went to the Congress and trying to negotiate with the NIH. So what Tony said in a way is I would say 99% of the diseases are correlated with aging, meaning when you're young, most likely you won't have them. When you're older, the risk is going significantly higher every year that you're older.
That's right.
And that's why aging is, some people like Nir Barzilai claiming that aging is actually a disease and we can prevent it. And some agree, some disagree, but we won't get into that today.
Yeah. I mean, my comment is, if aging is a disease, then life is a disease because as you just said, we're so tied to the aging process and you age at any time in your life, right? Your organism gets older, from the time you're conceived. And some of the cells that our body produces, they will never divide again. So we have them throughout our life. And, they somehow have to renew themselves, but they can do that less and less well. So, you can say it's a disease because the definition of a disease is not very well, you know, um, described. It's often, especially if you look at the component of aging in a disease, it's often very subtle at what stage an organ is not functioning well, and at what time you call it a disease. Alzheimer's disease is a perfect example. We know that the molecular changes in the brain start 20 years before you actually get clinical symptoms. And so, is that part of a disease, or is that part of the aging process? And it's a little bit of both, probably.
Yeah, absolutely.
It's a fascinating question.
Digging into that a little bit deeper, something else that you found was there's kind of 3 waves of aging that humans, I'm not sure if it's organism-wide or organ-specific experience.
Yeah, that is very strange, and but it's fascinating biology. So basically, what we did, again, we didn't have a question. We just used this method that allows us to measure thousands of different proteins, and we had over 4,000 people from age 20 to 90. They were all considered healthy. I mean, some of them probably have, especially in the older group, they may take medication for high blood pressure or, heart disease, but they're okay. They're not really sick. And when we measured these proteins, first of all, what we could see, what we just talked about, at any stage, in human lifespan, there are differences from younger people to older people. It continues to change. The composition of the blood continues to change. And so some proteins, they may be stable. So you have the same level for 50 years and then they go slowly up. Some might go slowly down. Other proteins, they go up throughout your life, sometimes in a very linear fashion, but more often they don't change in a straight line. And when we look at each age, so let's say we look at 40-year-old people and we ask, which of these 3,000 proteins we measured, which ones are higher or lower in 35-year people compared to 25-year-old people. So we make like a window across each age. And then we add the number of proteins that change at each given age. And what we found is we call it a wave of aging around 35 years of age, that a lot of proteins change significantly in composition. And what that means is people at age 35 have almost like an inflection point. A lot of biology seems to change from 35 to 40 compared to younger people. And we don't exactly know why that is, but it suggests, first of all, it makes again the point that, we age throughout lifespan, and we age not just when we typically think, old people, but there's a lot of changes that happen. And some of it has to do with, changes in sex hormones or menopause in women, for example, but men have these changes as well. So it's not just menopause. There are differences between men and women. But, what we speculate is that from an evolutionary point of view, humans lived probably around 35 or 30 to 40 years in the past before there was any medicines. There were always people who were resilient and escaped all the infections and all the diseases. And sometimes they would probably live to 100. And, there were these magic people who then often had, very big influence because they were so old, right? And they could tell stories about generations earlier, but they were very rare. The average lifespan was between 30 to 50 from all the records we have. And so this is almost like the natural lifespan of humans. And it's interesting that that's where we saw this very profound early wave of aging. Then we see another one at around 65, and then a big one at age 80 or so. And, traditionally people have studied aging more in these really old people and compared how are these old people different from young people. But what our research suggests is we should really start finding out what happens at this very early time point, because it's possible that this is when, your aging trajectory of a given tissue or organ starts to go in one way or another. Maybe you already are, sort of predestined to get heart disease at that very early age. And that's, of course, what we would like to know. The earlier we can predict, what happens to your body, the more likely we can intervene and work with an individual and give them advice of lifestyle changes or treatments that are already available.
Yeah, and Tony, I asked you before about, not ask, we discussed aging as a disease. The next question is also a bit theoretical question, but I think that's an important question. So do you think that the proteomic changes that you see are the cause or the effect of aging?
They're both. And we know this now because of these experiments that we did, where we exchanged the blood in mice from young to old. So, when we first had these findings that there are these dramatic changes in the composition of the blood, so how much of each protein is present in a young person compared to an older person, the question was exactly what you just asked. Is this just a result of aging? And so the blood basically just collects all the age information. And so the levels change. Or do these factors actually induce aging or contribute to aging? And there is sort of a somewhat macabre or, crazy experiment that people did, Paul Bert in Paris over 100 years ago, where they sutured animals together. And again, this sounds a bit gross, but they did this in part because they were interested in tissue rejection. And, they did all kinds of crazy stuff at that time. And they were just, I think, experimenting and seeing what they can do. Maybe also to see if they can, suture back on a tissue or a limb or, an arm or something in animals. So what they did in one of these models in mice is you basically make a cut in the flank of each mouse, and then you suture their skins together across the two animals. And because this is a wound, blood vessels grow into it, and then they actually fuse between the two animals. So now you have a network of blood vessels that allow exchange of blood from one animal to another. And again, that model was very instrumental, for example, to discover sex hormones, because if you had a male and a female mouse, you could actually see that you could induce changes, sex changes. It was also instrumental in understanding tissue rejection, transplantation of tissues, where people found that you can't just put a tissue from a liver, for example, from one person to another. They have to match for certain markers that we know now based on these studies. But this model allowed us to ask this question: what if we put a young mouse together with an old mouse? Can the young one make the old one younger? But also the other way around, does the old mouse potentially induce aging in the young one? And it's sort of fortuitous how we got to do this experiment, because my lab neighbor was Tom Rando. Unfortunately, he left now for UCLA, but and he also recruited me to Stanford. He had previously studied aging of the muscle, especially the stem cell of the muscle. So if mice get older, then the muscle does not regenerate as well. And also you can't build muscle mass. So if you're 80 years old and you go to the gym, your muscle will grow almost nothing. In fact, you often just cause sort of scars in your muscle tissue. And Tom asked the question, is this because the stem cells in that muscle are themselves aging, or is it something in the environment that they are missing? And by putting a young and an old mouse together, the blood from the young mouse could reach the stem cell in the old muscle. And he could show, I think in 2007, that this old muscle stem cell can be regenerated. And the muscle of the old mouse basically functions and regenerates like a young muscle, and also looked under the microscope like a young muscle again. And so he had this model that allowed us to ask the question, what is the effect on the brain? And what is the effect on the composition of these proteins in the blood? So what happens actually, if you put a young and an old together, the blood is not just the average of the two, but it is a mix of positive and negative changes. And that allowed us to identify then some of the factors that promote aging or promote rejuvenation.
Yeah. And just for our audience, it's really fascinating. It's called parabiosis. And I know that there are some clinic, I think mainly in California, maybe in Southern California, of some older people that receive blood from younger people. I'm not sure what is the scientific validity of that, but I know that there are some clinics that are doing that. And it's very interesting, but I'm not sure that there is enough data to support those treatment. But anyway, it's really fascinating field, and thank you so much, Tony, to describe it in a simple way for our audience. And Ashley, please continue.
Yeah, I've never heard of that other than a very scary movie that I don't even want to mention. Horror film from a few years ago where they sewed people together. I don't think it was for human health though. But of this, what's the effect on gene expression? So does it increase protein transcription or change?
Mhm.
Like, what causes some of these changes?
Yeah. So now we have tools where we can measure the levels of every gene. So when you go from basically the blueprint, the DNA, which is in the chromosomes, you first make a working copy. We call this an RNA or a transcript. And then from that, you make the protein. So we can now measure basically every one of these working copies, if you will, of these transcripts across the body of mice or humans. If we get tissue from humans, we can do that too. And we can use it, we can do it at an individual cell level. This is a technology that has completely revolutionized biology and medicine recently. And so when we use this technology, we can indeed show that every cell in the body of a mouse responds to either the young blood or the old blood. And some of these changes are entirely consistent with, in the young mouse, an acceleration of aging. So they basically mimic what you see in normal aging. And excitingly, in the old mice, some of the changes that young blood induces is really reversing these levels of gene expression, so that we call this rejuvenation, because it really looks like the tissue of a younger mouse at many different genes, and that's something that gives us, first of all, confidence that this somewhat crazy experiment is actually has a molecular basis that we can read out, and we can start to explain what really happens. And what I explained earlier with the experiment that Tom Rando has been doing, we can show that these stem cells, for example, they respond in very specific ways. And not just in the muscle, it turns out, but in the brain, stem cells respond as well. And also in other tissues. And there's now many different labs across the world who have repeated these studies and can show similar changes. And so it could potentially lead to the development of treatments. And, this, to come back to one of the first questions, this led to a founding of Alkahest, because we said, this is something that can be used potentially to discover positive or negative factors that can then be targeted therapeutically, or that you could use plasma or, the liquid part from donors, from human donors, to treat potentially diseases.
Yeah. So Tony, again, to try to clarify to our listener, because it's you explained it very well, but it's very complex subject, and also I have a question related. So let's assume that you take a 20 years old human and combine his blood using parabiosis to a 70 years old human. Okay. Let's assume that everything that you do in mice replicate to human. Okay. My understanding from you, the 20 years old human will behave like a 70 years old human, but the 70 years old will behave like 20 years old. And I would expect more like they will be in the between, and both of them will behave like 45 years. So can you a bit elaborate about that?
Yeah. So maybe I didn't say that right. So you actually do not end up in the middle. It is cell or organ specific where you have the most prominent effect. So we find, for example, in the liver, liver cells are very responsive to the rejuvenating effect. So in an old liver, the liver cells respond to young factors. But to use your analogy, so a 70-year-old would then become maybe 60 years old or 55 years old. And the 20-year-old would maybe get 30 years old.
Okay.
So it's not usually that it goes all the way to the other tissue, right? But one important thing is, and to also alleviate concerns here that we're trying to advocate suturing people together, what we found is, and this is really why I think people got very excited. We were actually the first to show that you don't need to suture animals together. You can just collect blood from young mice and then repeatedly infuse it into old mice, or the other way around. And moreover, what we showed is you don't need the whole blood. So for the listeners, when you look at blood, it's this red substance, and it's red, or red liquid, and it's red because of the red blood cells. But it also has all kinds of other immune cells in there. So when you spin the blood down, you end up with a liquid, and that liquid is called plasma. It's a yellowish liquid, and that's where we could show all these beneficial effects, or most of these beneficial effects or detrimental effects are. And we believe it's a large part of that is in the proteins that we can measure. Now, there's also other molecules in this plasma, such as lipids or DNA, cell-free RNA, and metabolites, sugars, all kinds of different things are in there. And we can't exclude any of them, that they might have a contribution. But we favor to study proteins because they can be easily manipulated, and medicine has figured out how to make them synthetically, so you could potentially make drugs.
Yeah.
That was going to be my question. Is the future, we're going to have blood banks and people donate, or are we going to be able to create some of these things synthetically too?
Yeah, that's a great question. So this is one reason why I think the potential to translate it is there, because, in medicine, we have used blood transfusions for decades, right? And they're totally life-saving. And what people found is that when you, again, there's two ways of blood transfusions. One is you have an individual donor and you transfer the entire blood with all the cells from one donor to another. That requires that you're matched by blood type. But if you take only the liquid, this plasma, then what the companies who make these products do is they can actually pool the plasma donation from 10,000 donors, let's say. And they make one big pool of these, and then they isolate different components. So one of the best product, the most valuable product they make is a fraction that contains antibodies. So these antibodies protect people who don't have a functioning immune system against infections. So almost anybody who had a bone marrow transplant or who had cancer and is immunosuppressed, they all get these immune fractions which contain antibodies from volunteer donors who give plasma. That allowed us, and this is how this company Grifols, who acquired Alkahest, got really interested in our research, because they have, through that process, they have all kinds of different fractions of this yellowish liquid. And so that allowed us to test which ones of these have positive effects in the mice. And we could indeed show that some of these fractions, improve cognitive function, memory function in mice. And some of these went now into clinical trials in patients with Alzheimer's disease and Parkinson's disease. So this is one possibility, that you directly take this liquid or parts of that liquid, components of that liquid, and make it into a drug. But maybe more realistically, for the future, we want to know what are the key components that are either detrimental so that we can inhibit them, or are beneficial so that we can make them synthetically. But we may need to combine multiple components. It's probably not just one protein. We're pretty sure it's not just one, but it's multiple different ones. So that provides or poses additional challenges, because our medical system, that regulates drug approval, has no good method to test the combination of multiple drugs, for example. Yeah. So it still provides a lot of challenges. It's very encouraging. The initial clinical trials with these fractions look promising. And in one trial, they actually treated over 400 people with Alzheimer's disease, and they had significant benefits in people with advanced disease, but it's still, not a final approval, and larger trials will be necessary.
So Tony, follow up to that. So if we are going back to the parabiosis, you suit the mice together and then you separate them. How long after
We actually don't separate them, but other people have done that. Yeah.
Okay, so let's assume that you separate them.
Yeah.
My question is, how long after separation would you still see the effect? Maybe to transform it to human. Let's assume that you inject plasma into their blood. How long will be the effect? Is it a short term? Is it a long term? Is it middle term?
Yeah, it's a combination of effects. So a really good study was published this year from James White and Vadim Gladyshev at Harvard. And they did exactly this experiment that you said. So they sutured young and old mice together, left them together for 3 months, and then they separated them. And actually, in one experiment, they let the mice live as long as they would live, right? They basically looked at mortality. And what they found is that the old mice that had been paired for 3 months with young mice lived significantly longer, several weeks longer, than the old mice that were paired with young mice. And so this is, 10, 15 months later, there was still an effect, on lifespan. And then they used molecular measurements. You may have heard about this epigenetic clock, where people measure changes in the DNA. So they use this to estimate the biological age. They use that to demonstrate that tissues in these old mice that were exposed to young blood were actually younger epigenetically. So to your question specifically, in our experiments where we infuse just plasma, what we can see is that a single infusion, for example, activates certain genes, even in your brain. So you give an intravenous infusion, just 5% or 10% of the total blood volume of the mouse, and you see immediate activation of certain genes that are known to be involved in functioning between neurons. Very quick effects. And then you see effects that last a bit longer, that would go for, days or weeks. And then you see some effects that last for months. And it also depends how often you treat. So we've figured out a paradigm where initially we gave every 3 weeks for 3 weeks, we gave infusions every 3 days for 3 weeks. So 10 infusions of 7% at the time of the total blood volume, because that's the maximum we're allowed to infuse in a mouse. And then at Alkaes, they actually figured out you can do just once a day for 5 days and you get similar benefits. And that's what they did then in the clinic. So people came to the clinic and were infused once a day for 5 days. And then in the clinical study, they came back again 3 months later to have another round of infusions. So, it's clinically doable. And I think they received maybe half a liter of infusion per time point. But, it's still early days to figure out what the best way is to do that.
Super interesting. Okay, one more side question just for my own personal interest.
Mm-hmm.
These 5,000-ish proteins that you are, intentionally looking for, do we know, like, if all of those proteins have function, or can we connect them to, of course they're in a specific tissue, but do we know what they do or why they're being produced more or less?
Yeah, that's a great question. And that's actually, So the short answer is we know for some proteins what they do, but for many we don't. And the 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 promise of this type of research is not just that you can monitor health or predict health and disease, but that you figure out, because you know which proteins are part of that model that predict, how old a tissue is or an organ is, that you actually discover biology and potential drug targets, right? So we find, for example, using this transfer of young blood into old, that the mitochondria are a key target of it's known that they're a key pathway of aging, but they're also a key target of young plasma rejuvenation. And so the mitochondria are basically the little power plants that are in the cell. They make energy that the cell can use for all its functions. And with age, the capacity of these little powerhouses, basically, or engines, decreases. And with this rejuvenation, we can make them younger again and functioning better. So that's an example of where we look in an unbiased way what changes in the cell with young blood exposure. And then we see, there's all these genes that are part of the mitochondria or mitochondrial function. And so using this same idea, we will find other biological pathways. Some of them are known and some of them may be unknown. And we may find proteins that nobody knew what they're doing. We find they change very dramatically. For example, if we get young blood into the liver, and so potentially this becomes a target for a new drug that can mimic these effects.
So Tony, I would like to switch gear back to the organ aging. And you show that in most people, only one organ show accelerated aging. And do you have an hypothesis why is it that only one organ and why not a few organs are accelerated aging for a person?
Yeah, that is bizarre. And we have thought very hard about whether this is some mistake that we made, but we can't really come up with an explanation that this could be an artifact. So to just take a step back, when we looked at, we had a total of about 7,000 people, not quite 7,000 people in this study. And when we looked in those who are healthy, about 3,800 individuals who are healthy, most of them are above age 50 in this study. And we looked at so we got blood from them and took the plasma, that liquid, measured 5,000 proteins. And that allowed us then to basically estimate the biological age of each of the organs. And when we did that, we find that in about 20% of the individuals, one organ is clearly older than the average. And we made sort of an artificial cutoff. If you have the average of the population, we said if you're 2 standard deviations above that, we call it an outlier or an extreme ager. There are a number of interesting observations we made. One is that, again, it's only about 1 in 5 people who have an extremely older organ. And when you say extreme, it's really 2 standard deviations. And we don't know, is that very bad or is that it's an arbitrary cutoff? Yeah. But we find very few people who have much younger organs. So if you have a younger organ, it's usually not more than 2 standard deviation younger. And maybe that suggests that, there's really no way that you can keep an organ young without, an artificial intervention. That, aging is relentless and you can only have faster aging organs or the same age organs within a certain range. But you find very few people who have clearly younger organs, extreme young organs by that definition. And then if you look now at those 20% of people who have extremely old organs, what's interesting, and this is what what Gil just said, is that they usually have only one organ is older and not multiple. And we don't know why this is. Maybe it is because we're really catching these very earliest changes. And, these people are by definition healthy. They're always in the control group of our studies. Yet, there could be underlying disease building up, and one of their organs is showing that. So we find, like 1% of the population or 1.5% of the population have older kidneys in this study. And a similar percentage have older hearts. And we can then show that these people who are extreme older ages, they have even a higher risk of developing that organ-specific disease. And they have a higher risk of dying also. And then we find about 1.5% of the population that we looked at who have multiple organs older. And we call these multi-organ agers. And these people have a 6.5-fold increased risk to die within 15 years. Wow. So this is again using the Barzilai study. And we did not have enough statistical power to make these calls earlier, because you can imagine, we would like to look at 100,000 people or 1 million people, right? Then we could make much stronger predictions even. Then we could look how many died within 5 years or 3 years. And if we then have the blood at time 0, we could potentially say your risk is 10-fold higher that you will be dying in 3 years, if all your organs are older. But again, we need more samples to make these claims and these predictions.
And Tony, you used the sample from part of the sample was acquired from Nir Barzilai that look at a long-lived human and control. Have you seen a difference in the proteomic age or the organ age between the centenarian versus the controls?
So we actually did not look at the centenarians in this study. We looked only at the younger population. So some of them, the offspring, so these would be the children who potentially have the same resilience to become very old because their parents got old, but then also their spouses who have not long-lived parents. But we are trying to do studies in collaboration with Nir and also another scientist in the Netherlands who has a study that is called the 100+ Amsterdam cohort. So she studies, Henne Holstege is her name, so she studies people who are 100 years old and still healthy, and then she follows them until they die. That's also a very interesting cohort. But yeah, we're absolutely interested in seeing whether they may have extremely young organs.
Yeah. It's interesting. Was there any individuals that had, organs that were more than 2 standard deviations older? You said there's siblings that are there sometimes. And again, maybe there's not enough power to see this, but
yeah, we don't have enough power. Yeah, so we'd love to do, of course, much more. At this point, we provide an example what one could potentially do, and I think we have pretty good evidence within the samples we've studied that we're correct, because we have 4 different groups of individuals. So we'd actually learn from one group, and then we test our discoveries in another group independently. This is called validation, independent validation. And we can get quite reproducible results this way. But again, it's only 6,500 people or so. And with all these studies, the more samples, the more people you study, the better your prediction gets, and the more likely it will hold up with all the details that you describe.
Incredibly interesting, and so much potential.
Yeah, there's a lot of interesting biology in there that I think we can follow up, and hopefully other people will start to use the same approach.
Okay, I've got another question of, you also found that adipose tissue is a really strong predictor for mortality. And we know that body fat distribution is pretty significantly different between males and females. More specifically, central or abdominal visceral body fat deposition correlates with increased susceptibility for metabolic complications, while more fat mass around the waist or hip area distribution usually is associated with reduced metabolic risk. And your measure of adipose, is it looking more broadly at total body adipose tissue, or basically showing that obesity across the board is predictive of mortality?
Yeah, we don't know actually. We don't have specific proteins. We haven't looked for specific proteins that would reflect one type of fat tissue versus another, because we only look, where does a protein potentially come from? And these are just proteins that we know are made uniquely in fat tissue. But that's a very interesting question. Maybe if we have more measurements, more proteins, some of them will come, from one type of fat tissue versus another, right? There's these white fat and gray fat or brown fat, that maybe there are specific protein markers for these.
Yeah. And talking about a different organ, that's basically the brain. So you found that a high number of proteins, I think that's around 200, that actually are derived from the brain, which is very interesting. And I think that it was much higher than any other organ. It was like literally the leader. So can you estimate or talk a bit about the accelerated brain aging and how it is defined, and also what is the effect of extreme brain ager on cognition?
Yeah. So what we were able to show is that the brain is actually the highest number of organ-specific proteins in the blood. And maybe this is because it's so highly specialized. In the brain, there's a lot of cell types or processes that are very unique to the brain, and they're not. These proteins are not used in any other part of the body. So we get, it's great that we get a lot of biological insight into the brain. What we find with these proteins is that they predict, as they change with age, they predict the relative age of the brain. And so again, people with older brains, they tend to show cognitive decline more likely over 5 years in our study than those who have younger brains. And we also find that patients with Alzheimer's disease have older brains than healthy controls. And that has actually been shown with other methods by other researchers in the past. So it's interesting that we can confirm that simply by looking at proteins in the blood. And for us, this is, as we mentioned earlier, my main interest is brain aging and neurodegeneration, and why does the brain become susceptible to, cognitive dysfunction in almost anybody if they live long enough. I mean, there are a few centenarians who seem to still be cognitively intact, but it's very rare. First of all, very few people live to 100. And of those who live to 100, a very small number really still has, cognition intact. And I think if you talk to older people, their biggest concern is that they lose, their cognitive function. And for us, it's super exciting, the possibility that we can read out molecular function in a way from the brain in a living person, right? These molecules, they come from the brain. These proteins, they come from the brain. So can they tell us very early how the brain functions and how it becomes potentially susceptible to Parkinson's disease, to Alzheimer's disease, or some of these other types of brain impairment? Frontotemporal dementia is another one.
As you said, these are giving you a printout of an active, alive brain. Because most research, we can't do, unfortunately, until someone passes away, and then you can research the physical brain.
That's right. Yeah.
You mentioned earlier of some of these proteins kind of tracking with someone before they have a diagnosis of dementia, but maybe they start to have some memory problems. Is there anything like that someone could use to identify if they themselves are an accelerated brain ager?
Yeah, so that's the idea of using this technology basically and validating it in more individuals, and then develop a test that you could administer or have administered, and probably with insight from a physician to help you interpret the data. Although some findings could probably be given to an individual directly. But I think the information is most useful. I mean, it's not useful to you if you know that your brain is 2 years older. So what are you going to do? You would really like to have the advice of an expert of telling you, for example, people who did this, they had a younger brain on average. People who did that, or people who used this supplement, showed, an improvement after 5 months of treatment or something like that, right? So as we collect more information and then also have information on interventions that people do, or even if you take a drug, if we measure somebody's brain age today and then they get one of these new antibody drug treatments, and we would measure their brain age in half a year or in a year again, and it shows, oh, this brain age is now younger, then you have a bit more confidence that, first of all, what we measure is probably true, but also that this drug is probably doing something. And so I think as we collect more information, we will be able to not just use the measurements that we make to make these predictions, but then also have useful interventions where we have hardcore data to support that, yes, this is reducing now your risk of getting a heart attack or getting cognitive decline.
Yeah, and related to that, Tony, you discuss interventions. So in our database, we see that sleep is a major intervention that help human to improve a lot of blood biomarkers. So I was wondering if you had that data on your cohort and you look at the effect of proper sleep on brain aging.
We don't. Maybe it exists from some individuals, but from the cohorts we have at this point, we did not use that in our assessments. But yeah, that's extremely interesting.
Yeah. Okay.
Well, this brings us to our final question, and this is something we ask all of our guests: as if there is a top tip that maybe you do in your own life that you've found in your research to improve health span. It's hard to narrow it down to one sometimes, but is there one thing that you'd be willing to share?
Yeah, it's pretty boring exercise. And, from our studies in animals, in mice also, we can exercise mice. We can take the blood of exercised mice and put it in non-exercise and basically make their brains function better. You have to be careful when you go from mice to humans, but in humans too, there's so much evidence that exercise is beneficial. So as long as you can exercise, that's definitely, the best advice. And then, I have a very, very diet, sort of Mediterranean-like diet.
So, Tony, what kind of exercise do you do?
I run, and I also do sort of Pilates yoga exercises.
I like your, I don't know if I like it, but I can see so many people are so resistant to exercise that now maybe the thought is, well, can't I just take the blood of someone that does exercise and get those benefits?
There is a study in Norway that is being done like that. Yeah.
That's funny. Thank you so much for everything that you shared and explained. I certainly learned a ton. Very much appreciate learning from you.
Yeah, thanks for having me.
Thank you so much. Yeah, that was an amazing discussion, and I think that our audience will like it a lot. Thank you so much for dedicating time from your busy day.
Thank you.
Thanks for listening to Longevity by Design. Please subscribe to this podcast on Apple, Spotify, or YouTube. Longevity by Design is powered by Inside Tracker, a personalized health optimization platform that helps people improve their lives by improving their bodies from the inside out using personalized, science-backed recommendations for nutrition, supplements, and lifestyle changes. To learn more, visit insidetracker.com/podcast.